myosin molecule. So each myosin head, I have kept the heads in red color. So each myosin head has two binding sites. So two here and two here. So one binding site is for the actin molecule and the other binding site is for ATP. Same here. One for actin, one for ATP. So the ATP binding site functions as a ATPase. So this one here, let's shade it. This one here, it functions as a ATPase. ATPase is an enzyme. So now let us continue with the myosin molecules. Each myosin filament is composed of more than 200 myosin molecules. The myosin molecules leaves the tail and extends towards the head in this portion. So this portion, let's call it the arm. So we have the arm and we have the head. The arm and the head together, the arm and the head together are called cross bridges. I repeat, the arm and the head together are called cross bridges. So each cross bridge, I repeat, each cross bridge is flexible at 2 points. The first point is where the arm leaves the body. So suppose this is the body here or the tail as we say it. So here at this point, let's shade that also. So at this point, the arm is leaving the body or the tail. So at this point, the arm is flexible. And also at the second point, the arm is flexible. The second point is here. This is the point where the head attaches to the arm. So at these 2 points, the cross bridges are flexible. The arm allows the head to move either forward or backward. The total length of each myosin filament is very uniform. It is exactly 1.6 micrometers. 1.6 micrometers. However, there are no cross bridges in the center of the myosin filament for a distance of 0.2 micrometers. Remember that. No cross bridges at the center of the myosin filament for a distance of about 0.2 micrometers. So, the myosin arm or head rotates to an angle of 120 degrees. Rotates to an angle of 120 degrees during a muscle contraction. Now let's see the actin head. I repeat, actin molecule. Now let us start and see the actin molecule. This is a wonderful diagram from a source book. So the actin molecule is actually a complex of 3 proteins. So let's call it an actin filament. So the backbone of the actin filament is a double-stranded F-actin molecule. So F-actin molecule is the one which is shown in 2 colored strands. This strand here and this strand here. So the strand are wound in a double helix in the same manner as a myosin molecule. So one strand of F-actin is actually composed of polymerized G-actin molecules. Attached to each one of the G-actin molecule is one molecule of ADP. ADP. The ADP molecules are the active sites on the actin filaments. The ADP molecules are the active sites on the actin filaments, where the cross bridges of the myosin filaments interact to cause a muscle contraction. Each actin filament is about 1 micrometer long. Remember that a myosin molecule or a myosin filament was 1.6 micrometers, whereas actin filament is only 1 micrometer. Let's go to the second molecule. Here it is. Here it is the tropomyosin molecule. The tropomyosin molecule is also wrapped spirally around the sides of the F-actin molecule. In the resting state, that is when the skeletal muscle is at rest, the tropomyosin molecule lies on top of the active sites of the actin strands. So when the muscle is at rest, the tropomyosin will cover the active site of the actin strand so that actin and myosin filaments cannot bind to each other. So tropomyosin covers the active sites on actin molecule. Now let us see the next one, that is the troponin complex. The troponin complex is called a complex because it is made up of 3 subunits. So these subunits are troponin I, troponin T, and troponin C. Troponin I has a strong affinity for actin. Troponin T has a strong affinity for tropomyosin, which is another protein. And troponin C, as the name suggests, has a affinity for calcium molecule. So this complex is believed to attach to the tropomyosin site to the actin. The strong affinity of this one, troponin C, is believed to initiate the muscle contraction.